bright and early, and now we're going to talk about bioinformatics, which normally might be a recipe to fall asleep, but I'm going to keep this practical, right? So I'm going to talk a little bit about how some of the tools work, but I'm going to try to frame this in terms of what would you want to do, why would you want to do it, and really importantly, not just like use tools blindly, but understand what the output really means, right? It's not really useful to get something that says, oh, this is a really great guide, and then you say, well, what does that mean relative to what and in what context? One thing that I really like to stress to people that are, not everybody is super comfortable with bioinformatics and computers, and one thing that I really like to stress to people like that is that bioinformatics really seriously is just stuff that you could do yourself, right? You could sit down with a piece of paper and a pen and do the types of things that these tools are doing. It's just that they're doing them really, really, really fast, right? So I think you'll see from the description of some of the way these tools work, it seems fast, it seems like magic, but it's just doing a lot of multiplication, for example, really, really quickly. So I think one of the fundamental things that I want to talk about today is the question of what makes various guides different. How do we know whether a guide is going to be active? How do we know whether a guide is going to be inactive? How do we know whether it's going to have a lot of off targets? And I think that is really borne out by these guides. So maybe those of you in the audience who are like super CRISPR aficionados can look at these sequences and tell me exactly what these are. But these are actually very, very commonly used sort of benchmark guides in the field. And they have very different properties that you might not be able to tell just from looking at the sequence. This is one of the EMX1 guides. It's very highly active. And it's known, based on some of the unbiased sequencing methods, to be relatively specific. This, on the other hand, is VEGF Site2, which is also one of the benchmark guides. It's also highly active, but it has hundreds and hundreds and hundreds of off targets in the genome, right? And just from looking at this, it might be hard to say, well, why is that? We'll talk about that in a bit. And this is one of the guides that we found while screening through guides for a biology project. And this guide is basically completely inactive. We've been unable to find any activity for this guide whatsoever in the gene that we've been looking at. And so what separates this guide from this guide, right? Why is one hugely active and the other one completely inactive? And that's part of what the bioinformatics software is trying to predict, right? So we want to gather huge amounts of data on guides like these that have various properties and try to collate that into some sort of predictive thing so that you don't have to be screening through lots and lots of guides all the time and say, is my guide active? Is my guide inactive? Oh, good, I found an active guide. Uh-oh, maybe it also hits 100 other things in the genome. So the general algorithm behind finding a guide goes like this. First, you find guides, and where you're going to find those guides, the region that you want to look for, really depends on your experiment. So you've heard about CRISPR cutting, you've heard about CRISPR-I, you've heard about CRISPR-A, and you've heard that there are different regions you might want to target. Even if you're doing, say, a cutting experiment, there are different places that you might want to target. Some people target, say, 5' regions of exons. Chris Vakach has a really nice paper showing that if you put guides next to, say, functionally important residues, then it doesn't really matter whether the indel is in frame or out of frame. It screws up the protein. So I think that there are a lot of different ways you can choose this. And I think there's no one good solution. It depends on the biology. It depends on what you're trying to do. Once you've found them, though, you then need to take each one of those and you want to map them to a reference genome. Okay? Reference genome, for example, for humans right now is called HG38. And these are just sort of the one true genome for humans. And one caveat that I really want to put forward for that is I think everybody in the audience would understand that your genome is different from the reference genome. And if you have a cell line, it's probably different from the reference genome. There are companies out there that are taking variant calls, SNPs that are found in cell lines and trying to produce custom guide finding algorithms that can map to custom cell lines. Desktop Genetics is basically making a service out of this where companies can go sequence their own cells of interest, their own lines of interest, send them to Desktop Genetics, and then you can custom find guides and custom do off targets with that. But it's a subtlety that most, at least academic, open source type software doesn't really take into account because it's a really hard thing, right? Lots and lots of variants. So keep that in mind as a caveat. Typically in our lab, what we do is if you have some site that you really, really, really care about, right? Let's say you're trying to do HDR in one particular region. You might sequence that region in your cell line of interest so that you know exactly what that region's sequence is in your cell line. because there are certainly trivial cases where your guide of interest that doesn't work because your cell line of interest has a SNP, say, in the PAM, and therefore you're not going to get any activity. So when you do this type of mapping, you want to know a couple of things. You want to know, where is my guide? Hopefully you knew that going into the experiment. You want to figure out where the potential off-targets are. You want to know if those off-target, basically where that guide would sit in those off-targets relative to genes, transcripts, and exons. Maybe if you have a highly prevalent off-target, but it sits in the middle of a gene desert, and you're just doing, you're not making a therapeutic, you're just doing research, maybe you don't really care. Maybe you can do a complementation experiment to take care of that. You also very frequently map these guys to other annotations, especially when you're trying to do sort of forward predictions. So let's say you have a huge data set. We have all these guides, and we want to know what works and what doesn't. We want to make some sort of forward predictor. You might try to correlate guide activity with, say, DNase hypersensitivity, how much that gene is transcribed, things like that. And people use these in so-called machine learning approaches to try to describe features that distinguish active guides from inactive guides. And then all of this kind of stuff gets put together into a score that you get. And so if you guys have used any of the CRISPR activity or off-target scores, you know that you get some number. It's very often reported between 0 to 100, but it's just an arbitrary score. And this usually takes into account a bunch of things about the guide itself, the sequence of the guide, as well as things about the genome, right? Does it sit in a hypersensitive site or not? Where is it relative to a transcription start site? Things like that. So we're going to go into these in detail for a couple of the most popular approaches. Starting off with finding guides, this is the really easy part, right? So it's the beginning of the morning, it's 8:30, everybody's tired, so let's start really easy, okay? Finding guides is just finding PAMs. And this gets back to the whole bioinformatics is just stuff you can do by hand, just does it really fast. Whenever you're doing guide finding, all you're doing is looking for PAMs. So in the case of pyogenes, that would be NGG. PAM can differ based on the organism, of course. And then you just count backwards 20 bases. So if you see something GG and GGG, right? So the first G, and that would be the NGG, you find a PAM. And then I also underlined this one. And just to wake people up, this is a really easy question. Remember, again, it's 8:30 in the morning. I'm not trying to throw you anything hard. Why did I underline this? Because it's not an NGG. Yes, it's the reverse complement, right? So obviously, don't forget about the reverse complement. If you forget about that, you're going to be missing half of your guides. No one has found any difference between the Watson and the Crick strand in terms of activity, for obvious reasons. So clearly, this guy, counting backwards 20 bases from here, just gives you this protospacer. So how do we map the guides? So let's say we have some region of interest. Maybe we have 100 bases, and we know we want to find guides in there. And we get a list of, say, I don't know, 10 guides. And we want to know where those map, somewhere inside our genome. We got this whole sea of a genome. If we're in humans, it's big. God forbid we're working in onions or something, right? It's even bigger. So where is this little tiny 20mer like? So if you think about this problem, it's actually really good that we live in the era of next generation sequencing, because not only do we have these genomes, if you think about this problem, you're trying to take a short sequence and you're trying to map it to a reference genome. And that should sound pretty familiar because that's basically short read alignment. And so the way people find a lot of where these guides go is they use short read alignment algorithms. So very popular ones have names like Bowtie, BWA. These are very often used by guide finding software, to map found guides back to the genome. We'll talk in a little bit about some of the caveats related to that. There is one difference here, which is called Cas-Off-Finder, which is from Jinsoo Kin's lab. This is an incredibly brute force mapping program. The only way that it actually works in like human time is by using GPU-enabled programming. But it does work. It takes a long time. But we'll get into why you might want to use Cas-Off-Finder instead of a tool that uses some of these other things in just a little bit. So now for each of our 10 guides, we know where they map. So these have coordinates that look like this. So for example, chromosome 8, and then it would be from this position to this position on the minus strand. Or chromosome 15 from this position to this position on the plus strand. Now we want to know what's there. And again, we live in the era of next-gen sequencing and ATAC-seq and DNA hypersensitivity mapping and all that stuff. So thankfully, we know a lot about what's in genomes. This is still an open question, and so you should recognize that there are a lot of various tools out there, Ensemble, UCSC Genome Browser, but you always want to take what's there with a grain of salt. So for example, knowing where transcripts are. Just thinking about where a transcription start site might be sounds like a very obvious thing, but there are active programs, active projects out there to try to better map transcription start sites. And you might imagine that if you're doing a CRISPR-I or CRISPR-A experiment, knowing exactly where that TSS is going to be quite important. If you're just trying to do exon mapping, that's maybe a little bit less important. We have a pretty good idea where exons are in the genome by now. But designing CRISPR-I, CRISPR-A guides can be a little bit more subtle. These types of websites have really nice interfaces. So, and they even have ways that you can upload browser tracks, so to speak. So you can take guides that you've mapped and you can upload them to these sites and see where they map relative to your gene of interest. Some of the guide finding and prediction software will actually do this for you. So it will map to various annotations. Choosing which database to use, Ensemble versus UCSC, these sorts of things, I consider that outside the scope of this workshop. Each database kind of has its own set of information and specializes in its own thing, whether it's transcription factor binding sites or epigenetic marks or things like that. So again, it comes back to what kind of biology you want to look at and what you're trying to do. One thing that I do want to point out, though, is really make sure that you're using consistent genome versions. So relatively recently, in the last couple of years, people have decided to switch to a new sort of consistent genome version, which is called HG38 or GRCH38. But there are still a lot of annotations in so-called GRCH37 or H19. And you just have to make really careful that, say, if you find guides in one genome version, you're not trying to map it onto a different genome version. Because you can imagine that, let's say, one genome version has this numbering. And then, OK, great, your guide sequence is still here. But a million bases upstream, something shifted by 1,000, right? You're going to be totally out of register for that. Your mapping is not going to work well. So just be aware that there is this sort of discrepancy in numbering that happens when you switch between genome versions. And just double check. If you find guides in one genome version, make sure you're doing mapping in the same genome version. OK. So the first big problem, we found guides. And we want to know which of these 10 guides is going to be the most active. So this is, there are a couple of really easy things here that are sometimes forgotten that I wanted to point out before we get to the more subtle approaches. The first one is if you're expressing a guide off of a plasmid, make sure that it does not have 4 T's in a row, because that's a terminator sequence. Okay? So if you try to make a guide off of a plasmid, not by IVT, which is what you're doing in the lab, if you try to make a guide off a plasmid and it contains 4 T's in a row, you'll never get that guide. It's not going to be transcribed. Okay? In general, you want to avoid homopolymers. So a variety of people have found that the more homopolymers you have in a row, the worse that is. And I mean, if you just think about that, skipping of polymerases, things like that. If you have a guide that's, say, 8 C's in a row, you can imagine you're probably going to have a bad time even transcribing that guide. People have figured out that GC content should be in a relatively reasonable range. So remember, Cas9 is all about bringing together guide RNA and your target. So if you have a really low GC content, the annealing of your guide RNA to your target is going to be compromised. Now, on the other side of things, people have started to notice that very, very high GC content guides tend to give a lot of off-targets. So I'm not going to talk about that data in any detail. But if you have a guide that is like 90% GC, it tends to have a lot of off-targets. And the thought is that you get inappropriate annealing because there's some sort of energy threshold to discriminating off-targets versus on-targets. And once you cross that threshold, it doesn't really matter whether you have perfect matches or not. Okay? You've just crossed the threshold. So if you have, say, a 95% GC guide and you still only annealed 17 of those bases, well, you still crossed that threshold. In general, I think at least that you want to avoid secondary structure. So this maybe has more to, I think there's maybe more data on this, even though it's anecdotal, from T7 transcription. So people have not seen a huge amount of correlation between guide activity and secondary structure for plasmid transfection when you're introducing guides on plasmid. But in our lab, we have certainly seen that when you try to use T7 transcription to make guides that have a huge amount of secondary structure, you get a lot of inappropriate truncation of those guides. You get all kinds of weird stuff on a gel. You guys are going to be running RNA gels to see these things. They run really strangely on the gel. Cas9, to some extent, is probably going to be able to take care of that because it does have, the protein has energy to be able to remove secondary structure and enforce the type of, the binding to the secondary structure that it wants, but we don't really understand the details of that. So in general, you would want to avoid the following. So remember, there's a constant region. Generally, we try to make sure there's relatively little structure in the protospacer. You don't want some sort of really strong stem loop in there. We also try to make sure that the protospacer doesn't interfere with the constant region hugely. But these are kind of rules of thumb so far. So this is not something where we spend a huge amount of time thinking about it and looking at it. We might run it through a secondary structure prediction server like Vienna and just say, is it completely ridiculous? And if it's not completely ridiculous, still go ahead. OK. Surely it's more complicated than these little tiny rules. So one really nice set of work coming from John Dench and David Root, which you guys are probably familiar with, looks at maybe active guides have certain sequences. And so for this, I'm going to be giving these slides out, so you don't have to write this whole URL down, but there's a really nice server that goes over some of these tools. So the experiment here is we want to figure out maybe some guides, we know for a fact that some guides are more active than others. And the question is, why are some guides active and some guides are inactive? So the experiment is, first, we're going to take a gene. It's got a bunch of exons. And we're just going to tile over everything. Exons, introns, everything. We're going to do this for a couple of genes. Make every single guide. Okay? Then we're going to make pooled libraries, pooled lentiviral libraries. And these are cell surface proteins, so we can just sort for loss of protein. Okay? If a protein goes away, we can say, oh, this guide must have been active. And then if you just sequence what comes out of those cells, maybe you could tell the difference between an inactive and an active guide. And this seemed to work OK. So this, on the y-axis, would be fold enrichment, so sort of a corollary for activity. So things that are high up here are good guides. Things that are down are bad guides. And you can see that there are guides in, say, the first exon that have a range of activities. In the third exon, there's an even bigger range of activities. A non-essential exon sort of falls out. So that's a nice internal control. And so now the question is, can we take this and turn this into some sort of score? But there's a catch. And the catch is, if you do the exact same experiment in different cell lines, you can actually get very different activities between the guides. So in some cases, things match up really well. So things that are active in multiple cell lines. Sorry, to explain this, the y-axis here is one cell line. The x-axis is a different cell line. These dots are the relative activities of different guides in each cell line. And now, here, this is plotting the same guides just against a third cell line. And you can see that these two cell lines correlate with each other really well. And these cell lines, things are pretty good, but they also fall apart a little bit. There are some guides that are really active in one cell line and basically completely inactive in the other cell line. And you can also get vice versa, right? This guy has an activity of almost 100% in this cell line and only about 20% in this cell line. So guide activity can be cell line dependent. Local sequence effects might be a problem. We don't really know the sequences of these lines. Maybe there's something about SNPs in there. But let's try to average it out, because there is a pretty good correlation going on here. So if you take all of those guides across multiple genes, and you say, what makes a good guide and what makes a bad guide? And then just look at the sequences, you can get some kind of patterns out of this. The first thing that comes up really strongly right here is that right next to the PAM, if you have a G in position 20, that's really good. And if you have a C there, that seems to be relatively disfavored. But obviously, all this other stuff going back behind here is pretty complex. And so the idea is we don't want to, every time we have a guide, we don't want to be going back and being like, oh, I have an A there, but what if I had a G? And that's a little bit worse. And, right, we want to have some sort of computational predictor that can be trained on all this data. So the way this was done is you take each base at every position in a 30mer, so it includes sort of a window outside the protospacer and outside the PAM. You turn it into a feature. So you say, what if there's an A here? What if it's a G here? What if it's and then you go to the next base. What if it's A? What if it's G? If it's T? And then you even do combinations of bases. So you do, say, the dimers. There's also features for GC content. So you take all of these features, and you put them all together, and you tell a machine learning program, here's my data of good guides versus bad guides. And these guides have various features, right? One feature might be this G, and one feature might be this C, and one feature might be this T. Then you ask the machine learning algorithm, tell me which features are the most important. That is, which ones have high weights and which ones have low weights. Okay? But we need some way to tell it what's good and what's bad, because we can't just give it all the data. We have to sort of usher the computer through the process. And so given this kind of observation that there's correlation in some ways and correlation not other ways, they decided to say, what separates the top 25% guides from all others? Okay, so you take the top quartile. And what separates the top quartile from everything else? And that becomes important in a little bit, as we'll see. Okay, so you got your best 25 guides, or your best 25% guides, and then everything else. What separates them? And so you use machine learning to find the sequence preferences. This has been called rule set 1. And we'll see in a little bit, there's now a rule set 2, but it's pretty similar. One thing that I want to point out is that machine learning is weird, right? Machine learning is not about finding rational rules for stuff. Machine learning is about finding weights that just give you the optimum predictor of the data. And so things don't have to make physical sense. They don't have to make any sense whatsoever. They just have to be able to predict the data that you gave it. Okay? And so, for example, in this set, there are some properties that are really good. So for example, if you, in the weights that they found, some of the best properties are a GC at this position. For some reason, if you have this dimer, the machine learning thinks that's a really good idea. That ends up actually being different in rule set 2 when you add more data. Conversely, putting a GC right here, it really hates it. It says, oh, you should never have a GC at that position. And again, when it comes to machine learning approaches, you should never try to rationalize these things. It doesn't have to do, again, with like what Cas9 likes. You can try to interpret that way. But it's really, you're just asking an algorithm to say, I have a set of data, and tell me what feature weights will give me the closest agreement to the data that I gave you. So it's not a bad thing. It's just a caveat you have to remember, right? What that means, though, is when you do this type of training, right, this is the data. Since you're training on this data, the features that you get are going to look exactly like that. It's going to do well in back prediction. That is, if you try to go back and you try to predict stuff that you've trained on or even stuff that you've left away, it does really well. And in this case, it seems to call good guides pretty often, 50 to 75% of the time. But there are some caveats to this, which is when you're doing machine learning does really well on things that you've trained on. So remember, it's a cutting data set. It's scanning across exons. It's looking in a certain number of cell lines. And since we're not talking about any kind of like fundamental insights into the way Cas9 works, training works best in the way that you have trained it on. Okay? So you have to remember that this data set is looking at cutting and knockout. And so it's actually not totally clear that training using this kind of training set for, say, CRISPR-I or CRISPR-A would actually be applicable. Because one of the major features in actually the next rule set in 2 years afterwards that John Dinch's group came up with was proximity to the 5' region of the gene. So whether you're in the first exon or not. Okay? That may be very relevant for a cutting type of screen. We know that for CRISPR-I and CRISPR-A, it's relative to the TSS. But for this rule set 2, the, in fact, major feature that they found was where it was inside the coding region. Okay? So clearly that rule, which is default in rule set 2, doesn't have a lot to do with CRISPR-I, CRISPR-A. So all I'm saying with this is use tools for what they're meant for, and don't try to cross-mix tools, especially if machine learning was used for that kind of data generation. So what's rule set 2? Rule set 2 was really just doing their first paper, but doing it on steroids. So they decided to take a bit more data, and they decided to use more complex machine learning. They had a guy from Microsoft come in who was a machine learning expert, and he threw a whole bunch of different algorithms, and they kind of did a little bit better than the previous one, but maybe not quite as much. And then they decided to add a lot more features. So they've got, I think they had like 500, almost 600 features that they decided to add. The thing to notice from this graph is that most features don't matter at all. The y-axis here is the contribution to the weight. So they added in a huge number of features, and most features don't really do anything. Very common in machine learning, because the way you train machine learning data is you come up with everything you could ever think about and throw it in, and then the computer will tell you what's important and what's not. Okay? So what you can see are a couple of features matter a lot, and then there's a very fast drop off. Yeah?
So I can imagine different nucleotide combinations that would constitute all of those features. How do you come up with over 500 different features at a position? What are examples of the software features?
Yeah, so they started doing dimers that are not next to each other, and trimers, and just like all kinds of stuff. But the things that most came out of this, so that's all in this long tail, right? The stuff that most came out of this, again, is proximity to the 5' end of the gene. So if you're trying to knock out a gene, you want to put it in the first exon. Okay? The TM. So you want to have the TM between the guide and the DNA be in some reasonable range. Like we talked about before with GC content, not too high, not too low. That's a very strongly trained feature. The single and the double base features, just like in the first rule set, ended up being very important. And then again, this G at position 20 seemed to be very important. So one thing that I want to point out about this is that, so rule set 2 is actually, it's very powerful. It's very predictive. It works very, very well for knockout. So I certainly don't want to say that this is not a good thing, because in my opinion, if you're trying to do knockout, you should be scoring your guides based on this rule set. It can be very, very powerful. But don't mix apples and oranges, is all I'm trying to say. So again, this is where you would find this. I've been saying this over and over again. Use the tool for what it's for. So this is a way to find active knockout guides. It's trained on knockout by single indels. It was trained on mouse and human cell lines. And it was also trained on cell surface expression and survival. And I think these are important caveats. And you'll see why in a second. So these tools find highly active guides for knockout pretty well. I would say actually with rule set 2, it does really, really well. But remember what I said earlier about this quartile. Okay? I mentioned that, and I told you it would be coming back. And here's where it comes back. When you train the data on the top quartile, you're saying, tell me what the top 25% of guides look like. And then differentiate them from everything else. And that means the trainer is going to try to differentiate a top 25% from a 50%. Okay? So the algorithm will tell you that a 50% guide is going to get a poor score, because there's a differentiation between the top quartile and that 50%. Okay? So these tools will report false negatives. So they will tell you that a guide that's, say, 50% active is going to look relatively inactive, because of this whole trying to find just the best guides and scrunch things up. Okay? So just keep that in mind. If your guide that you find experimentally works pretty well, 50%, let's say, and the tool says, oh, that should be a bad guide, don't freak out and say, oh my gosh, this tool is bad. That's just the way it works, right? It's not trying to do what you're talking about. One of the important things to remember about this tool is that a lot of these features are sequence-based. And so they do assume that there's one true guide sequence, that is Cas9 is going to love a certain protospacer, right? That it has protospacer preferences. And I think that's actually important to point out. I mean, these tools do work quite well, but the underlying assumption is that Cas9 prefers different protospacers. And that is not totally clear from some of the biochemistry. So I think that these tools work, but as you'll see, there are other things that can go into guide activity besides just protospacer sequence. And like I've been saying all along, this type of software is probably not going to be very good for making guides for CRISPR-I, CRISPR-A. Doesn't mean that you shouldn't try it, right? It can certainly, because of this preference for TM, the features for TM, to avoid homopolymers, things like that, it will let you avoid some of the worst stuff, right? But if you find a guide that works, that this tool predicts is going to work really well for cutting, and it does not work for CRISPR-I and CRISPR-A, again, it wasn't made to do that. One thing that's really great about this tool is that the code is on the website. And so if you have someone in your lab, or if you yourself are a bioinformatician, and you want to make sure that this thing actually works the way that it's advertised to do, you can go and you can look at it, and you can convince yourself. And in fact, if you have new ideas for things to do, you can add those in. And that's actually, I think, very, very important, especially in a research environment. So like I've been saying, based on this whole idea of one true sequence, you might think that Cas9 has certain sequence preferences. But in our lab, we certainly think that Cas9 is going to prefer certain sequences, perhaps, say, right next to the PAM. But we don't think that this is the whole story, and this is why. So at the same time that one group used human and mouse lines to come up with this rule set, coincidentally, another group did basically the exact same thing in zebrafish, right? Hundreds, thousands of guides trying to look at knockout. And very conveniently for us, they plotted their data in almost the exact same way. So this is the data in zebrafish, and this is the data in human and mouse. And some of these guys are very similar. So the colors are a little bit different. But again, Cas9 prefers very certain sequences. So for example, a C in zebrafish is really strongly disfavored, and a G in zebrafish is really strongly favored. And in human and mouse, C disfavored, G disfavored. So great. But you can see that back here, a lot of the preferences kind of seem to fall apart. And if you think back to some of the original data on correlation between guides, there were some guides that work really well in one line and some guides that don't work well in another line. And so why might this be? Okay, why would you get, say, something that's really active here and not active here? And so just as a teaser for a paper that we have, it's in press right now, we found out that you can actually take any given guide sequence and you can make it better. Okay, so you can take a poorly active guide, and by messing with DNA repair outcomes, you can turn it into a really, really active guide. And the secret here is actually adding non-homologous oligonucleotides. We think it soaks up a DNA repair factor. So we just use, say, a BFP sequence. And you can turn, say, 20% guides into 70% guides just by adding these guides in. Okay? So you'll take guides that the guide RNA prediction software says these are terrible guides. And in fact, they are terrible. But if you mess around with DNA repair, they become good. So through this and a couple of other things, we have an idea that there's sort of this round-robin cycle. If Cas9 comes in, it'll find a sequence, it'll cut it. There's some fraction of the time that you get error-free repair. The site is just completely put back together. And then Cas9 will come in again, and it will cut it. And the way that you exit out of this is through the error-prone repair that we tend to see, indels, right? And so this is the exit from the cycle. It's sort of the trap. And so we're either through adding in these oligos, we're either inhibiting error-free repair or activating the cell line-dependent activity. And so one thing that comes to mind is that Cas9 may have certain preferences. Remember this GC next to the protospacer or next to the PAM. But some amount of sequence preference that you see that's different between cell lines may have to do with the ways that different cell lines will repair different sequence contexts. So there's sort of this mystery is the wrong word. There's sort of this hubbub floating around the genome editing literature right now about microhomologies. What is the role of microhomology-mediated end joining in the indels that we see? Sometimes people see indels, this type of error-prone repair, that really strongly correlates with microhomologies. And sometimes people don't see that. And it's sort of unclear at the moment to what extent sequence context is really going to matter. So I don't have anything to say definitively in terms of pathways or anything like that. But I think that it is possible that, like we've seen here, if you were to say run the dense root algorithm, human and mouse, and you yourself were working in zebrafish, you are probably going to have a bad day because those trainers are not meant for your data. I just want to spend a minute or two talking about finding active CRISPR-I, CRISPR-A guides. I don't want to spend basically any time on this because I think you've probably already heard this story and you'll probably hear much more and in much better detail from Martin. But this was done in a way kind of similar to what you heard about from Denshin root, which is you just tile. So you take some sort of transcription start site, you tile around it, and you look for things that say, oh, this is going to be a very active CRISPR inhibition, CRISPR-I region, or what would be a CRISPR-A, CRISPR activation region. And there are certain regions around the transcription start site that seem to correlate with better activity. And you'll probably hear more from Martin about their really exciting recent work on even better predictors and better correlation for CRISPR-I and CRISPR-A. So there are a lot of other features you can look at. There are relatively few online resources for finding CRISPR-I, CRISPR-A guides. There is one from Stanley Chi's lab called CRISPR-Era. This will do cutting, inhibition, activation. I don't think that it contains a lot of the newest stuff from Martin, Max, Luke. But I'm sure that's probably in the works. OK, so we talked a lot about activity. Before I move on to specificity, do people have questions about activity? Yeah?
Is there a way to train the software to look at RNA binding protein sites in these regions? And do you think that can affect the guide activity?
That RNA binding sites, so you're thinking about like the guide RNA gets soaked up by some sort of factor in the cell? Is that?
Yeah, or there are regions which are more amenable to splicing factor binding, or say some of the ribosomal RNA.
Oh, I see what you're saying. Yeah, no, that's,
Binding protein.
yep, that's absolutely possible. So some people have gone back and tried to take some of this sequence-based activity and then layer genome annotation on top of that. And as you might hear from Martin, that does seem to be the case for some things. Maybe I won't spoil the thunder, but there are certain annotations that correlate really well with binding sites, or there are certain activities that correlate really well with binding sites in the genome. That's for CRISPR-I, CRISPR-A. People have not found that so far for CRISPR cutting. But if you think about it, that's probably because people are mostly looking in bodies of exons. They're not looking around exon-intron junctions or anything like that. They're looking in the bodies of exons. And so usually those regions are pretty free and clear because, especially in highly transcribed genes, polymerases are rocketing through them anyway. Other questions about activity? OK. Oh, yeah.
Just does TA modification do anything to the Cas9 activities of the actor?
People have looked at this in vitro. Oh, yeah, sorry. The question was about DNA modifications. So people have looked at methylation of DNA, things like that. They have not found that that has an effect. So that's actually a nice feature of this particular system. OK, so moving on to specificity. So of course, we want guides to be really highly active. But if they're really highly active and they chop the genome into 1,000 pieces because they also cut in 1,000 other places, that's not going to be super useful for our experiment. So how do we know whether or not a guide is going to be specific? So just like with activity, there are some really easy things to avoid. Number one is repeat sequences. So if you have a sign or a line element or something like that, you probably don't want to put a guide in that. That's not going to be a good time. But it's easy to forget about these things. So if you just look at, say, genome browsers, things like that, they will often identify repeat sequences via upper or lowercase letters using programs like Repeat Masker. And you just want to make sure that your guides don't fall into these places that are highly repetitive in the genome. Like I said earlier, so Chris Vekach has said, so you want to target things that are conserved for function. But the flip side of that is if you take a highly conserved part of a gene and it's represented in another member of a gene family, like let's say you have a DNA sequence that's, maybe you have a bromodomain sequence and it happens to be in a bunch of other highly conserved bromodomains, and you target that region, you're probably going to target every single one of those bromodomains. So you want to avoid that. But again, is it more complicated? Probably most people in the audience are familiar with this server. It's the CRISPR MIT server. This is one of the first prediction servers that was out there. And it's based on really great data from Patrick Hsu in his paper in Nature Biotech in 2013. So this is really around target-focused specificity measurements. So you could either make one guide and then find all the targets for that. And that might be a guide-seek or a BLESS type experiment. But for this, what they did was they made mini guides and measured the activity at one target. So in this case, they targeted EMX1 in 3 different places, and they just stepped through every single possible base at every single possible position. And they asked which ones still target EMX1. Okay? So the general idea is we've got one target. We're going to make all the possible variants of guides that target this one target and which ones still work. And you end up with these beautiful maps that look like this. So here, activity is in dark blue. On the x-axis are various mismatches. Sorry, on the y-axis are mismatches. On the x-axis are positions along the protospacer. The PAM would be over here. And so you can see that there are not only position-dependent propensities, but also some mismatches are better tolerated than others. So for example, this GT mismatch seems to be pretty well tolerated throughout most of the sequence. But then the CC mismatch can be tolerated out here, but is really unhappy next to the PAM. So what they did was they took all of these features and they said, this is kind of complicated. Let's collapse this down into one predictor of mismatch. Okay? And this gives you kind of 2 general areas of a protospacer. There's the so-called seed region, in which you can have mismatches that completely kill the activity, and the non-seed region, in which mismatches are relatively tolerated. The definition of what's seed and non-seed kind of differs. So some people say that the seed versus non-seed starts here. And you can kind of see why, right? There's maybe for this UC and the CC, there's a little bit more tolerance there. So usually when people write down what's seed and what's non-seed, they would say like between these bases and these bases, because no one has really defined what exactly seed versus non-seed means. Because again, this is not based on any sort of physical description of Cas9 recognizing something. This has to do with just sort of phenomenological data about what do we see from a genome editing experiment.
I hate that distinction.
Seed versus non-seed? Yeah.
So if you go back one slide, if there's a CC mismatch way off at the left end, way outside the seed region, but it can make a big difference.
Mm-hmm.
And in the guide seed type data, you find quite a number of significant secondary targets that have mismatches only outside what people call the seed region. So I think it sends you in the wrong direction.
I would completely agree. Yeah, I would completely agree. But I want to make sure that if you guys hear, it's very common in the literature for people to talk about the seed versus the non-seed. I want to make sure you guys know what they're talking about when they talk about it. And where does that data come from?
And to add that, I think that concept comes really from RNAi, where there is actually more of a meaning to the seed, because the seed is really the region that can mediate microRNA-like
Right.
translational silencing, right? So there is a structural significance to it, and I think they apply the concept here. But
Right. Yeah, I think it's sort of stretching the analogy a little. It's a little bit far. Yeah.
And I have to add that comment. I think that's exactly right. How much of this is just human nature? We're already comfortable with the concept of a seed and we want so badly to apply it to something that isn't useful. If everyone is doing this work in order to make genome editing more successful and accessible to a broader community, are these kinds of exceptions really confounding and dangerous?
I don't know if I would say they're dangerous. I mean, one thing that I want to point out about off targets, and I should have said this at the beginning. When we talk about off targets, I think for most of the people in the room, and probably even for people that are doing clinical work, I think that off targets in some ways have been maybe overhyped in the literature, and I think dangerously so. So I think people are really obsessed right now with finding all the off targets, predicting all the off targets, doing a huge amount of sequencing to find all the off targets. If you think about it, if you're in research mode and you want to make a knockout, if you were to come to me and say, oh, I computationally predicted and there are no off targets, I would say that's great, but show me the data anyway, right? You'd want to do a complementation experiment. You'd want to make another clone. It's the same thing that you would do with an shRNA or an RNAi experiment. Just because it's CRISPR doesn't exempt you from having to do good controls, right? So I think in that way, yeah, you don't want to pick something that, say, is a known repeat sequence and is going to hit hundreds of times in the genome. But even if you do have some off targets and you can show that the phenotype can be complemented by re-expressing your gene, you're probably fine. Even in therapeutic mode, I think that there's this big debate roiling right now around off targets. And personally, my feeling about off targets for therapeutics is that the most important thing is to know about them and to be able to deal with them appropriately. So just to give some context to this, so I was a group leader at Genentech for several years, working on drug discovery projects, working on therapeutic projects. And no small molecule is ever completely clean. No antibody is ever completely clean. It's impossible, right? The important thing, though, is that you have a functional assay for, say, genotoxicity, for safety. If you have a completely efficacious genome editing reagent, and it is completely 100% specific, it could still have on-target toxicity, right? So the important thing is from a therapeutic point of view, having a functional measure of off-targets, basically. So I think that on a sequence level, it's something that it's important for us to think about. It's teaching us a lot about how Cas9 works, and it's letting us avoid making silly mistakes. I think that some of these patterns are still useful for helping us design our experiments, but we should not take them in any sort of way as like, oh my gosh, I have 3 off targets. I should never use this guide. I should never do this experiment. Or, as a reviewer, if there are any people reviewing papers in the audience, if someone shows that a guide has off target, that's not something where you say, oh, this is junk, right? Reject out of hand, right? This is, of course, they want to be doing controls, things like that. The point of off targets is not that they're off targets. The point of off targets is that we need to know that they exist, and we need to know how to do the right controls to think about them. So that was a little bit of an aside about off targets, but it's something that I feel strongly about. Okay, so getting back to the data. So there are some interesting patterns for some of these off targets. So it has been seen that if you take mismatches, and if you make these mismatches really close to each other, that seems to have a stronger effect on activity than if the mismatches are far apart. So if you have 2 mismatches next to each other, that really hurts guide activity. Whereas if you have 2 mismatches, the same mismatches, further apart, it's more permissive. And this, even though this data is sort of, it's looking at in vivo activity, so it's separated from the biochemistry, it does make a lot of sense in the context of the biochemistry. So through careful biophysics, we now know that what Cas9 does is it takes the PAM, it cracks open around that PAM, and then you get invasion of the guide RNA around that, okay? Sort of going down the line. And you can imagine that if you have 2 mismatches in the row, that's going to be a pretty high bump for that invasion, the strand invasion, to get over. Whereas if you have 2 mismatches separate, now you just have 2 smaller bumps to get over and perfect annealing in between, okay? So this type of data makes perfect sense in light of what we know physically about Cas9. So now we want to take all of this, and we want to turn it into some sort of score that we can use computationally. And so how do we do that? So I didn't want to put this equation up until, now it's 9:30. I didn't want to put it up right at 8:30 because I think these types of equations scare people usually. But it's actually really easy, okay? So these are trained, again, they're weights that are trained from that heat map that I talked about. The training details in the paper are a little bit unclear, and they've actually changed between versions of the website. So I think of it as like a work in progress. What does this mean? For those of you that don't do a lot of math, this is basically just the same as sigma, which is adding up terms, but instead you multiply them. You look at products. So you look at 1 minus the weight at the position, and then you multiply by this thing that looks really complicated. We'll get into what that complicated thing is in a moment. These are the weights that they originally trained on. You can see again that the weight is not very high, pretty far away. So that means 1 minus 0 is still 1, so you get a really high weight there. Whereas out here, there's a big penalty, so 1 minus a big penalty gives you a smaller number, and then you're multiplying everything together. What are these? This penalizes mismatches that are close together. So this is just a heuristic term, so to speak. So there's no sort of theory behind this. They just chose an equation that would give you something based on a distance, and this penalty goes up if you have mismatches that are close to each other. And this penalty goes up as the total number of mismatches increases by the square of the number of mismatches. So what does this look like? So if you had a mismatch here, that's 3n, 0.014. So 1 minus 0.014 would be the weight. Okay? And everything else is a perfect match. So you do 1 minus 0.014 times this stuff. And if you have another one here, you'd have 1 minus 0.685 times all this stuff. Okay? Now let's say you have two mismatches. You'd have 1 minus this position 0.8, where'd it go? 828 times 1 minus 0.685. Okay? So you see this total weight goes down as you get more and more and more mismatches. Does that make sense? I go over this in some detail because I think a lot of people sort of use the CRISPR MIT server as this like black box of like, I get specificity out of it. And I think it's really important to know where the data came from and how it's getting these scores, because the scores are on just this totally arbitrary scale. So is this pretty clear? Okay. So that's for one mismatch. But obviously, there are all sorts of other sites that you might find. Okay? So what we need to do is we need to do an aggregate score for that guide across all possible sites. So you score it across all the off targets. How a sequence is chosen hasn't been documented, so it's closed source. We don't know how it's finding stuff. But it has become clear recently that it's using bowtie 2. And I point that out for a reason, and we'll get back to that in a second. So all you do is, for every single one of the possible mismatches, you aggregate them together and you normalize them to 100. So now you get some score between 0 and 100. If you have a lot of high scoring off targets, that's going to decrease your score far below 100. If you have off targets and exons, this web server will note them, but you don't get any sort of extra penalty or anything like that. So having an off target in a gene desert versus an exon is counted the same. So that's for off targets. We can get even fancier than that. So in the same time that Dench did the big rule set 2, they also looked at on-target stuff. And this was it was a pretty neat experiment. So they took 28,000 guide RNAs targeting CD33. And these 28,000 guide RNAs are every single on-target guide RNA. They made every single one-base bulge, so not a mismatch, but trying to make bulges in the RNA, bulges in the DNA. And then they did also all the mismatches. And they made that in a lentiviral library, and they did the same thing that they did for the on-target score, right? Sorting for CD33 surface expression. And this is what the data looks like when you sort for that. Okay? So this is the perfect match, and you can see that you get a nice distribution between a log 2-fold change of 2 to 4. And these are, in black, the variance on the perfect score. So you can see that some of these variants look exactly the same as the on-target. And some of them look the same as the negative controls. And then there's stuff kind of in between. So they ended up turning that into a heat map. I think this heat map is it's a little funny. So it's a lot of data. It's much more data than Patrick Hsu's stuff. The reason I think it's a little funny is that it's just a reordering of this exact same heat map. So if you sort of do the mental gymnastics and you take these various rows and you reorder them to look the same as this one, the data is exactly the same. Okay? So looking at 28,000 guide RNAs, they got the same answer that Patrick did by looking at EMX1, which gives you a lot of confidence, right? These do seem to be pretty strong preferences that Cas9 has for discriminating different types of bases. This got put into something called the CFD. Remember, this also incorporates bulges, though, which Patrick Hsu's score does not incorporate. Okay, so there is a little bit more discrimination there. So this is called the CFD, and this is also available on that website that was on one of the previous slides. So now, of course, we have to ask, how well do these things work? Okay, and there are various ways that people have come up with to, in an unbiased way, find off targets. They have names like guide, seek, and bless. And you might ask yourself, how well do they correlate? If we predict something is going to be off target, how well does that correlate with being off target? And at first, things look really bad. So for example, the MIT server predicts a lot of off targets, and guide-seek finds relatively few, and there's not a lot of overlap, right? VEGF-Sci2, all the way across here. MIT is predicting a lot. We're not actually finding that much. Well, again, the point of the MIT server and the point of the Hsu stuff was to not say these are the off targets, but to say these are the possible off targets, right? These are the things you should be worried about. So personally, I think we shouldn't be that worried about the fact that there's this big orange thing. I would actually want a computational tool to tell me, oh, you should be worried about these things even if they're not real. Because personally, I would like to at least know about something rather than have something be false reported or reported negative. The thing that I would be worried about is the stuff in this blue, which is stuff that the servers do not report but are actually off targets. So there's lots of room for improvement. And what's wrong? And we actually got the answer to this in a paper that was published, I think, 2 weeks to a month ago. So this is from, gosh, I can't remember the last author of this. It's early in the morning, I guess, and so I'm blanking. So what they did was they looked at correlation coefficient between known, like a real off-target and a false off-target, between the CFD, between the original SHU score, and this thing called CC-top. CC-top is really fast and easy, and that's why some people use it. And they saw that if there were a few mismatches, there was pretty good correlation for everything. And then as the number of mismatches increased, it went down really fast. And then they got a big surprise. So this dark blue line is the off-target score from the SHU website. Okay? If you recalculate the score using the published algorithm, and they found their own off-targets, they instead got this purple line. So using the exact same score, they got different answers. Why would that be? And the answer is because the MIT website is using Bowtie 2. And these guys instead use Cas-Off Finder. So remember, the score is the same, but you're aggregating across all of the possible off-targets. And what they found is that Bowtie actually has some bugs in it. And so Bowtie has trouble even finding off-targets as the number of mismatches increases, because it wasn't built for this, right? It's a short read alignment. Okay? If you instead use this Cas-Off Finder, which remember I told you before is like this ridiculous, incredibly exhaustive thing to find every single possible off-target, then you get something that's really, really good. So the exact same score used on two different sets of potential off-targets gives you different answers. And in fact, almost completely overlaps with the stuff that you see by the unbiased sequencing. So the problem here is not the score. The problem has nothing to do with choosing seed versus non-seed or any of this kind of like sequence preference. It has to do with how do I build my list of things that might even be off targets to score. Okay? So it's a non-trivial problem because there's a lot of 20mers in the genome that you would have to evaluate. And so things like Bowtie or BWA take shortcuts to make that happen in a reasonable amount of time. If you do it really, really brute force, you can get it to work. And so maybe there's something in between, and this is, it's an area that people are talking about right now.
But they still underperform.
Sorry, that was a lot of questions.
They still underperform.
Compared to CFD, yeah, they do. Yeah, so this CFD is done using Cas-Off Finder. So they do underperform, and I think they underperform because CFD, the main difference here, this gap, is bulges. Yeah, so remember, SHU score is not looking at bulges, it's only looking at mismatches.
And also just to add to that, for people who use BWA type algorithms or Bowtie themselves, there are parameters that will find everything, right? They just slow down those programs, but it's not an actual problem with the BWA algorithm. It doesn't take
Very,
in the sense that
very
you
true.
can't find them. It's just which parameters you set for the search, right? So you
Yeah.
can use those tools to find everything.
Absolutely, yeah, absolutely true. So a lot of these programs, they have a lot of command line parameters. You have to tweak these things, and the types of parameters that you might use for, say, short read alignment for sequencing might be different than what you might use for a 20mer. And so we shouldn't be surprised that you might get things underperforming if you just try to use it straight out of the box.
You say this Cas-Off Finder is really slow because I think it's not feasible to do for full libraries of off targets. You have to restrict like maybe it's feasible if I want this one gene knockout, I just want to make a library of guides. Is that possible, or is that software way too slow to do that?
So it kind of depends on what kind of setup you have, right? So it uses a GPU. So if you've got, if you're just trying to use your laptop's GPU to do this, it's not going to work, right? If you're just trying to use even, say a Mac Pro GPU, it's not going to work, right? But you can buy GPU boxes, right? Which are multiple GPUs. Those can work. And so So in the lab, we have used Cas-Off Finder to make limited libraries. It takes much longer to make those libraries and to score all the off targets. But as long as you have the right hardware, it's totally doable. Yep.
So the Keith Young experiment is set up in a way that it's sequencing between 10 and 50,000 genomes per experiment. And that strongly argues that the amount of information on true off-targets that are validated by genome sequencing is vastly underestimated because per gene, they undersample the potential of off-targets. These experiments are designed to highlight the existence of off-targets.
Yes.
But none of these experiments are designed to exhaustively. So my main criticism of all of this is that at the end of the day, you're standing with a number, and you get no guidelines for your actual experiment. Which of the 500 potential off-targets predicted do I actually have to look at? Right? I have to look at all of them. So there is no way out here. I really don't see it.
Well, I completely agree. And so the other thing that I would point out, I'm going to go back to all the way back to this oligo thing. So this thing that we call NOE, which is the enhancement of genome editing activity. The way we do this is by throwing in a non-homologous oligonucleotide. And that should sound kind of familiar to you because that is a guide-seq experiment. So a guide-seq experiment is you take a non-homologous piece of DNA and you throw it into your genome editing experiment. So what we've seen is that just the act of putting in a guide-seq oligo or any type of non-homologous oligo boosts apparent editing at both on and off-target sites. And so I think that there are certainly cases where you would get something that's way down in the weeds. And by guide-seq, it's going to show up, right? So I think that following up from that, one thing that we've been doing in the lab is making custom capture libraries. So the same way you would do exome capture, you say maybe you would do an unbiased off-target. You get a list of a couple hundred or 1,000 or whatever things from guide-seq or BLESS or any of these methods, and then you make capture libraries. And then you really deeply sequence those capture libraries, and you say how many of those are actually real. And from the capture, there's actually pretty good algorithms based on variant calling, of course, to be able to quantify how much those are actually there. I think for one thing that I would point out, and I think this is partly your point, Dirk, is that from a therapeutic point of view, when we start talking about off-targets, in some ways, these sequencing approaches don't really mean anything, right? Let's say you want to do editing for hematopoietic disease, and you need to edit 10 to the 6 cells, okay? So you're going to edit those cells, and you're going to try to find some off-target. So let's say 1 cell or 5 cells have that off-target, okay? And you're going to put those back in. Let's say it's a really bad off-target. You can't sequence deeply enough to see a 5 in 10 to the 6th event by NGS, because the error rate of NGS is above that threshold. You can't do it, okay? So this gets back to the point that I made previously about functional tests for genotoxicity, functional tests for safety. There's no way that we can see the things that might be dangerous. So we have to design appropriate functional assays to look for them. There's just no way around it. I know you weren't expecting to be in a bioinformatics lecture and have someone tell you that you should be doing more experiments rather than bioinformatics. Okay. So I just want to spend a couple of minutes pointing out some of the really easily accessible tools that people might use. So this is the CRISPR MIT input. So you can choose genomes. You can give it sequences. This is limited to about 250 bases that you can search. And you get some really nice output out of this. So this would be your guide that you're interested in. Here are the potential off-targets, where the mismatches are, whether they're in genes. It looks really nice. There are other tools. And these are so-called heuristic tools. And heuristic basically means people come up with arbitrary rules. And all I want to say about those is that arbitrary rules doesn't necessarily mean bad. Just because something wasn't machine trained, doesn't have machine learning, doesn't mean that it's bad. So for example, this thing called ChopChop has a bunch of various heuristics. You get all these various arbitrary penalties. But these penalties kind of mirror what people have been seeing by experiment. And so the idea is whether machine learning told you, oh, this penalty should be 0.56 versus someone says, oh, I'm going to give this a penalty of 500, does it really matter? It probably doesn't matter. One nice thing about ChopChop that you guys might like is that it gives you really nice access to designing T71 primers. So when you put in your sequence, you can then choose the region that you want to target, and then it will auto-design primers for you. So it makes it really easy, a really simple workflow to go through. Another one that people have started using quite rapidly or quite recently is called CCTop. CCTop it's not one of the highest performing ones, but it's super, super, super fast. And this sort of gets to this whole idea that I talked about where you should just be using functional assays to validate, right, to do complementation, things like that. Because the idea is, you want to be throwing away the worst guides. You don't really care about things down in the weeds. You just want to get a feel for things. And so in that case, better to be fast and pretty good, throw away the worst stuff, and then it doesn't matter whether you can discriminate, the top 1 versus the top 2. You just need to be doing the experiment at the end of the day. Again, CCTop has a pretty nice interface for working through some of this stuff. This is Cas Offfinder. So Cas Offfinder is available on the web. You can search a variety of genomes for off targets. You don't give it sequences to target. You instead give it guide RNAs. So that's an important discrimination, right? You don't say, I have this region. I want to find guide RNAs. Show me where the off targets are. You instead give it a list of guide RNAs that you want to target. And there's a lot of other tools. Okay? So I don't want to exhaustively go through this. I just wanted to introduce you to a couple of them. There's one more that I wanted to show. So I'm going to show one of these, but I just wanted to point out 2 of them. People have started to aggregate a lot of these scores. I talked about a lot of algorithms. People have started to aggregate them in various ways. So there's a commercial offering free for academics from Desktop Genetics. They actually have this really interesting tool now which recalculates almost every score and will then show you all these multi-dimensional plots of how they compare to each other based on these calculated scores. And it becomes incredibly clear that all these tools come up with wildly different scores for this exact same guide. So it's kind of fun to look at it and just be like, well, OK, I shouldn't pay too much attention to this. Okay? I do want to show you really quickly benchling just to take you through a really fast workflow. I do this not as any sort of advertisement for benchling, but just because it shows you what you might do to design guides in your own hands. So has anyone in the lab used benchling before? Some people? OK, so benchling is the software that it's open source. You can do a lot of things for alignment, stuff like that. They've incorporated guide design. So what you see here is a sequence. There's an exon. This is a gene called UCHL5. You see there's a coding region down here. Let's say I want to design guides in this first exon. You would go over here, and then there's this tiny little button. You click to design and analyze guides. You tell it you want a single guide. You tell it what genome. Remember, I talked about the importance of genome preference or choosing the right genome, making sure that everything is consistent. You choose your region. You would say, oh, I want to find these things. You found it. And then it says for every guide, it gives you the sequence of the PAM, and it gives you an on-target score and an off-target score. So this is just a way of pointing out, this is this is sort of the outline of what I talked about in the beginning, right? What do? You find guides, you look for on-targets, you look for off-targets, you put that together, and then at some point, it's up to you as a human being to make the choice. Where do I want to use? What's the balance I want to take? And always take these scores with a big grain of salt. Anybody have any questions about that? That's all that I wanted to talk about for today. Yeah?
Question about your work that you said. When you put in your Innovative work, is that what it is?
Yeah.
When you put in your exogenous non-homologous sequence, is that length dependent or length critical, or is it need to be linear? And what are the rules that kind of govern that?
Yeah, so we found that, so it has to be above a certain length, but if you protect the DNA, then it can be shorter. So if you make it too short, but it's unprotected, it gets degraded in the cell, right? If it's protected, you can make something shorter. So we just use 120mer, has a BFP sequence. It's not present anywhere else in the genome. But we found that you get the exact same effect even if it's homologous. So the way that we actually discovered this was by doing a negative control experiment where we tried to introduce a silent mutation. And we found that our rates of indels skyrocketed when we were introducing silent mutations, together with also making the silent mutations. So there's that. You do need ends. So closed circular plasmid does not do it. The composition doesn't matter. You can throw in salmon sperm DNA, and it'll do it. It does, so the activity is cell independent. So almost every cell line that we've tried will boost activity when you do this. But the outcome of what happens is highly cell independent. So if you take these oligos and you put them into HEC293s, then when you end up getting is blunt end insertion of sequence. So the cell will start grabbing sequence from all over the place, any duplex DNA that has ends, and it will jam it into the break made by Cas9. In U2S, on the other hand, U2S does not do that. And instead, when you add in these N oligos, it makes large deletions. And I think this is because, of course, cancer cell lines are cancer cell lines. They have lesions in various repair pathways. They have differential expression of different repair genes. And so I think we can't expect every cancer line to be the same. In our hands, at least, most primary cells seem to act more like the K562s, HEC293s, in that they jam stuff into junctions.
This is great. Thank you. Great. Exactly what we needed for all these tools. Can
Thanks.
you give a comment on where the field's at for non-NGG band utilizations with the scoring?
Yeah, so the question is, where is the field for non-NGG and PAM usage? So a lot of these tools, you can choose alternative PAMs, all that kind of stuff. All they are doing is they're taking all the rules that people came up with for strep pyogenes Cas9 and just porting it over. So for the most part, they have not done the experiment again with the other Cas9. They're just assuming that it's going to work pretty much the same. Which I think is why you don't see these tools for things like CPF1, right? So I think people were comfortable with that assumption for, say, Strep aureus Cas9, and so they kind of ported that stuff over. But no one has made any tool like this for CPF1 because it's not clear that it's going to work the same way.
And how about for pyogenes Cas9 using non-NGG?
Oh yeah, so sorry, that was your question. So your question was more about use of NAG PAMs and NGA PAMs. Yeah, you can specify other PAMs. There are actually some really nice papers where people go through this type of work using NGA PAMs and NAG PAMs, yeah, and quantify exactly how much off-target activity you get at those sites. You know, what's the effect of having an NGA PAM and a mismatch, things like that. Those haven't been, so a lot of these servers have not been updated in terms of their underlying algorithms or training sets in a while. And so that stuff is not necessarily incorporated in here. So what you'd want to do in that case would be maybe get an initial idea of your guides with one of these tools, and then go back to some of the papers and try to weigh it against that primary data.
Just to clarify, in the transition from the efficiency to specificity, basically, but you said the broad site basically incorporates both in the end, because they also did, or at least in the paper they looked at both, or no?
They're separate, so they're separate servers. So the MIT server looks at the at Patrick Hsu's score from the Nature Biotech 2013 paper, and then there's a separate site on the Broad that is this sgRNA designer, which looks at rule set 2 and this thing called CFD.
Okay.
Right?
But with the CFD, it incorporates some.
Yeah. Yeah, exactly. So CFD is the specificity score, and rule set 2 is the on-target score.
But then you want to do something separately then for the off-target, or no? Basically, the specificity is.
Well, the CFD is the off-target score. Yeah, exactly. And CFD seems to perform pretty well in human cells. Again, this is all very highly machine-learned trained stuff, and so the same caveats that we talked about for activity apply, right? If you're training off targets on a certain gene for knockout in human cells, if you're doing an experiment in human cells, if you're working in human cells, you're doing knockout, you're probably pretty safe. But if you now move to Drosophila, right, you may be okay, but there's no guarantees made, right? And you shouldn't say, oh, no, I can't use it. But you should just have in the back of your mind, I'm going to use this tool, but I need to keep aware that this tool was not made for what I'm using it for.
How much of off-target is because of guide sequence or because of the Cas9 ability to be presented itself? Some of the experiments it's not possible to have transient transfections if we are expressing with viruses. So what would be a perfect approach in those conditions where Cas9 is present for a longer period of time?
Yeah, so I think that is a great question. So the question is about how long Cas9 is around. So if you remember, I'm going to go back to this slide. So if we think about this model where Cas9 is going around this cycle, if Cas9 is around for a long time, you would then expect it to have more and more and more chances to give you this error-prone repair. And that is exactly what we see. So in general, if you deliver Cas9 via an RNP, it's around for a very short time, and you tend to have fewer off-targets than when you're doing, say, lentiviral transduction or plasmid delivery. So the question is, in cases where you can't keep that time down, where you can't have this narrow time window of sort of, let's make our cut and then get out, what do you do? People have tried doing things like attaching Degron sequences, PES sequences, to Cas9, oxygen-inducible Degron, shield, that kind of stuff. The caveat, the problem, of course, being that Cas9 is already pretty big, and there's a packaging limit for AAV, and so when you start putting these sequences on, it gets tough. So most of the translational work that I know using AAV is just using stock Cas9, either SA or pyogenes. I'm not aware of people using any of these degradation sequences, at least translational yet, but I think it's a good opportunity to try some of those things.